Okay, so let's break down the phases of meiosis. We talked about previously how meiosis consists of 2 complete rounds of cell division, meiosis 1 and meiosis 2, and each round has 4 phases. Just like mitosis, it's going to have prophase, metaphase, anaphase, and telophase. So in total, meiosis has 8 named stages: prophase 1, metaphase 1, anaphase 1, telophase 1, and then round 2, which is prophase 2, metaphase 2, anaphase 2, telophase 2. So let's compare meiosis with mitosis to kind of understand how these processes are different. So meiosis is going to involve 2 nuclear divisions, where mitosis only has 1. That's a key difference. Meiosis ends with 4 daughter nuclei, and after the cell divides completely through cytokinesis, you get 4 daughter cells. In contrast, mitosis plus cytokinesis gives you just 2 daughter cells. Another big difference lies in the chromosome number and genetic identity. So after meiosis, the daughter cells are haploid, meaning they all have a half number of chromosomes as the original parent. So these cells are also genetically different from each other and from the parent. And again, why are they genetically different? Well, that's because we had crossing over and chromosome shuffling that we talked about. So on the other side, we have mitosis, which produces diploid cells with the exact same chromosome number and genetic makeup as the original cell. So let's look a little bit more at some of the differences between meiosis 1 and mitosis. So in prophase 1 of meiosis, something special happens. We mentioned it previously. We have synapsis, where homologous chromosomes pair up and form tetrads. You do not see this in mitosis. Then, during metaphase 1, there's tetrads lying along the equator of the spindle with homologous chromosomes facing opposite poles. So each tetrad has 4 chromatids total. In mitosis, chromosomes, which are referred to as dyads, line up individually, not in pairs. Then comes anaphase 1, where we have homologous chromosomes separate, not the sister chromatids. So that's different from mitosis, where we have sister chromatids that separate at this point and then head to opposite poles. Now for meiosis 2, which is described as basically the same as we saw previously in mitosis, but with one twist. So the cells going into meiosis 2 are haploid, not diploid like in mitosis. So while the mechanics are the same, chromosomes line up, sister chromatids separate, and so on. The genetic context is going to be different. Finally, just a little note about when and where these processes begin or are happening. Mitosis is everywhere, so it's occurring in all of our tissues, especially during embryonic development, and it's going to continue throughout life for growth and repair. Meiosis, however, is a little bit more exclusive. It only happens during certain stages of the life cycle of sexually reproducing organisms, and only in specialized reproductive tissues, like the testes and ovaries in humans.